Right now, as you listen to this, there is an organism actively rewriting the human genome.
Yeah, it's pretty wild to think about.
It really is. I mean, it has killed more humans than literally any war in recorded history. And it doesn't operate on this, simple linear battlefield where a pathogen enters, your immune system reacts, and we just give you a magic billet.
Right, we all kind of grew up with those military metaphors for infectious disease, right? Like invading germs and defending white blood cells.
Exactly, yeah.
Yeah.
But when you step into the world of global public health and you face a disease like malaria, that whole two-dimensional battlefield just vanishes.
It's totally inadequate.
What you're actually looking at is, well, it's the ultimate ecological chess match. And this is a game with 4 highly active, relentlessly mutating players. You've got a parasitic agent, a human host, a mosquito vector, and a shifting environment.
And every single time one player makes a move, like, say, a genetic mutation in our red blood cells, or we introduce a new pharmaceutical, it forces an immediate, crazy, complex adaptation from the other 3.
Which fundamentally changes how we have to study it, right?
Yeah.
So for those of you listening, whether you're a medical student staring down board exams, or maybe a junior doctor managing funeral travelers, or a public health professional trying to actually build systemic interventions, the goal today is to master the conceptual frameworks behind this chess match.
Absolutely. We are unpacking the core concepts from Chapter 8.15 of the Oxford Textbook of Global Public Health.
But, we aren't just going to sit here and summarize facts. You can read the text for that.
Right. This deep dive is about building the clinical and epidemiological scaffolding. Like, if you just memorize that an Anopheles mosquito transmits Plasmodium, you hold a piece of trivia.
Yeah, good for Jeopardy, not much else.
Exactly.
Yeah.
But if you understand the precise, mathematically predictable resting and feeding behaviors of that specific mosquito, and how the parasite's life cycle ruthlessly exploits human red blood cell genetics.
When you actually hold a framework. And in public health, frameworks are what save lives.
So true.
But we need to grasp the sheer gravity of the board we're playing on before we even look at the pieces. Malaria is arguably the most prolific killer of humans in our evolutionary history.
Currently, 99 countries report ongoing transmission. That's massive.
40% of the global population lives their entire lives exposed to a disease that is fundamentally preventable.
Right. And it's ancient. The Greeks wrote about these deadly cyclical fevers, but the players were totally invisible until 1880.
Right. When Alphonse Laveran, this French army physician, finally peered through a microscope in North Africa and literally saw the parasite thrashing inside a human red blood cell.
Yeah. And then it took nearly 20 more years of just agonizing meticulous entomological tracking before Ronald Ross, a British physician in India, identified the dapple-winged Anopheles mosquito as the actual vector in 1897.
Finding the parasite and the vector earned both of them Nobel Prizes, which really speaks to how monumental it was to finally just see the pieces on the board.
We finally knew who was moving against us.
Exactly. So to understand the interventions we use today, we have to interrogate the biological criminal at the center of this whole crisis, the agent.
The Plasmodium genus. And it's a massive genus.
Right, over 100 distinct species, infecting reptiles, birds, mammals. But human public health is really just consumed by 5.
Plasmodium falciparum, Plasmodium vivax, Plasmodium ovale, Plasmodium malaria, and the relatively newly recognized zoonotic threat, Plasmodium knowlesi.
So let me ask you, because when you look at high-level public health data, there's a huge temptation to just lump it all together, right? Like, malaria is malaria.
Sure.
Yeah.
A patient comes in with a cyclical fever, a headache, muscle aches, and you treat them for a parasitic infection. Why do we care so much about differentiating these species right off the bat?
Because differentiating them at a microscopic level isn't just some academic exercise in taxonomy. It dictates the entire clinical trajectory.
Like life or death trajectory.
Literally. If you misidentify the species, the patient can die, or they can suffer a totally debilitating relapse a year later. The species dictates the speed of replication, the severity of the organ damage, and the exact pharmacological strategy you have to use.
Okay, so let's break them down. Falciparum is the big one, right?
Yeah, Plasmodium falciparum is the apex predator here. It is the dominant driver of global mortality, responsible for the vast majority of severe complications and deaths.
Because it just replicates so fast.
It replicates relentlessly, yeah, and it possesses these unique mechanisms to evade the spleen, which we'll dig into later.
Okay, so that's the killer. But then you have Plasmodium vivax, and its mortality rate is much lower, which I guess historically made the global health community sort of underappreciate its impact.
Yeah, for a long time. Yet it has a massive geographic distribution, way wider than falciparum, and it possesses a biological weapon that falciparum lacks.
Hibernation thing.
Exactly, the ability to just hibernate in the body, and that hibernation completely changes the treatment paradigm.
Because if you just treat a Vivax infection with a drug that only clears the bloodstream,
The patient will literally walk out of your clinic feeling totally cured, only to suffer a massive relapse months later because the parasite was quietly sleeping in their liver the whole time.
that is terrifying.
Then you have oval and malaria, which share some similarities with Vivax but have much more limited geographic footprints. They generally cause less severe disease.
Right. And then there's the fifth player, which is just wild because it shatters the traditional human mosquito-human cycle. Plasmodium knowlesi.
Yeah, the zoonotic one.
Right. Traditionally known as a parasite that infected like long-tailed macaques in the jungle canopies of Southeast Asia. But recently it's surged as a major threat to humans, right? Especially in Malaysia.
Yeah, and the clinical danger with Nalesi is its replication speed. Like, Nalesi operates on a blistering 24-hour asexual replication cycle in the blood.
Wow. So every 24 hours.
The parasite load in their bloodstream literally doubles every single day. A mild fever can escalate into overwhelming fatal hyperparasitemia before the lab has even finalized the smear.
That's incredibly fast. Which means we really need to understand these replication timelines. You mentioned the liver, the bloodstream, the 24-hour cycles. Let's map out the biological path.
The life cycle framework.
Exactly, tracing it from the moment of exposure straight through to the physiological mechanism.
The easiest way to visualize this is through a dual host framework. Because the parasite is an obligate organism, it requires both a human and a mosquito to complete its genetic destiny.
Okay, so let's start with the human phase, which to me seems fundamentally about stealth entry, followed by just massive amplification.
That's a great way to put it. It begins when an infected female Anopheles mosquito takes a blood meal and injects her saliva.
Right, along with anticoagulants she's dropping in the payload.
Exactly. She inoculates this highly motile, microscopic form of the parasite called sporozoites directly into the human dermis and bloodstream.
And the fascinating thing about these sporozoites is that they don't just hang out in the blood, do they?
No, not at all. Because the immune system is patrolling the bloodstream, looking for invaders. The sporozoites basically know this. They act like biological fugitives.
So they run.
Within 30 to 60 minutes, they completely clear out of the peripheral circulation and invade a hepatocyte's liver cells.
Okay, so this is the exoerythrocytic schizogony phase.
Yeah.
Exo meaning outside the red blood cell.
Right. Inside the liver, they are effectively invisible to the host's primary immune defenses. They spend the next week or two feeding on the liver cell and undergoing massive asexual replication.
So one sporozoid goes in.
And a single one can multiply into tens of thousands of merozoites, packed tightly inside a structure called a schizont.
It's literally like a sleeper cell preparing for a coordinated strike.
Yeah.
Now, you mentioned the hibernation weapon of Vivax earlier.
Yeah.
Is this where that happens?
Yes, this is the crucial divergence. In falciparum, malaria, and Nalesi, every single parasite in the liver continues to replicate until the liver cell ruptures.
But not Vivax.
Right. In Plasmodium vivax and oval, a subset of the sporozoites enter the liver and simply stop developing. They enter a state of suspended animation.
Hypnozoites.
We call them hypnozoites, yeah. They're biological sleeper agents waiting for some unknown chemical trigger.
So a patient could, like, move from an endemic jungle in Brazil to a snowy city in Norway, get 0 mosquito bites.
Yeah, and a year later, that hypnozoite wakes up, begins dividing, and triggers a full-blown malarial crisis.
That's crazy. Which is why public health surveillance for Vivax really has to account for historical travel, not just recent exposure.
Exactly. Now, for the parasites that don't hibernate, the ones making those massive schizonts, the liver cell eventually just physically ruptures. It bursts, pouring tens of thousands of merozoites directly into the bloodstream.
And that triggers the second human phase, right? Erythrocytic schizogony.
Yes. This is where the clinical alarms finally sound.
The merozoites flood the bloodstream, but again, they don't just stay floating in the plasma. They rapidly attach to and invade human red blood cells.
And once they're inside, they mature through several stages. If you look at a blood smear, you first see this classic ring stage, which matures into a trophozoite, and then finally into an erythrocytic schizont.
So the parasite is literally eating the hemoglobin inside the red blood cell to fuel its own reproduction.
Yes. And eventually, it consumes so much of the cell and multiplies into so many new merozoites, anywhere from 8 to 32, depending on the species, that the red blood cell just violently ruptures.
unleashing the new merozoites to invade fresh red blood cells, and the cycle repeats. But we have to talk about the synchronization of this rupture, because that dictates the classic clinical presentation of malaria, right?
Right. They don't just pop randomly like popcorn.
Yeah. They somehow synchronize their biological clocks. When they burst out of the red blood cells, they do it en masse.
Which dumps massive amounts of parasite debris, malarial pigment, and pyrogens into the bloodstream, all at exactly the same time.
And the human immune system sees this massive surge of garbage and just freaks out, right?
It responds with a violent cytokine storm, specifically an explosion of tumor necrosis factor alpha. And that cytokine storm is what causes the hallmark malarial fever paroxysm.
The bone-rattling chills, the spiking fever, the profuse sweating.
Exactly. And because the parasites are synchronized, these fevers occur in highly predictable cycles dictated by the species' replication speed.
So for falciparum, vivax, and ovale, the red blood cells rupture every 48 hours, right? Causing a tertian fever.
Yep. For malaria, it takes 72 hours, causing a quartan fever. And for Anopheles, as we noted, it's that daily 24-hour cycle.
So the human is experiencing these rolling waves of severe illness. Their red blood cells are being decimated. Parasite numbers are exploding. But the parasite's goal isn't actually to kill the human, is it?
No, a dead host is a dead end. Its goal is to escape and infect someone else.
So how does it make the jump back into a mosquito?
It requires a biological pivot. Most merozoites continue the endless cycle of bursting red blood cells, but a tiny fraction of them respond to environmental stressors and alter their genetic expression.
So they stop reproducing asexually.
Right. They differentiate into male and female sexual forms called microgametocytes and macrogametocytes. And these gametocytes do not rupture the red blood cell.
They just sit there.
They just float in the peripheral circulation, waiting.
Waiting for a female Anopheles to
Yeah.
take a blood meal. And when she bites, she inadvertently sucks up these gametocytes. And what happens inside the mosquito's gut is fascinating.
Because the temperature drops from the warm human body to the cooler ambient air, and the pH changes. This triggers the gametocytes to break out of the red blood cells.
It initiates the sporogonic cycle.
Exactly. Inside the mosquito's midgut, the male microgamete fertilizes the female macrogamete, creating a zygote. This zygote elongates into this highly motile form called an eukaryote.
And the eukaryote literally burrows through the wall of the mosquito's stomach, right?
Yes, and it forms a cyst, an oocyst, on the exterior surface of the gut. Over the next week to 2 weeks, that oocyst undergoes multiple rounds of division, producing thousands of thread-like sporozoites.
And then the cyst bursts.
The oocyst bursts. These sporozoites migrate through the mosquito's body cavity, and they specifically zero in on her salivary glands.
Wow. And once they're packed into the salivary glands, the mosquito is officially infectious. But the timeline here is the critical vulnerability in the entire transmission chain, isn't it?
It absolutely is. The biological process I just described, from the mosquito ingesting the gametocytes to the sporozoites reaching the salivary glands, takes about 10 to 18 days.
Which is known as the extrinsic incubation period.
And that 10 to 18 days is the Achilles heel of malaria, because in the wild, a mosquito's life is brutal and short.
Right, they don't live very long.
The statistical probability of a mosquito surviving 18 days to actually become infectious is quite low. If she's eaten by a bat, killed by a spider, or swatted by a human on day 12, the parasite dies with her.
The transmission chain is permanently severed.
This is why, mathematically, mosquito longevity is the single most powerful variable in the epidemiology of malaria.
So if the survival of the insect is the ultimate vulnerability, we really need to transition away from the microscopic parasite and look closely at the insect itself. We need to define vector ecology.
The focus here is entirely on the female Anopheles mosquito. Male mosquitoes are biologically incapable of transmitting the disease.
Because they just drink nectar, right?
Right. Their mouth parts aren't designed to pierce human skin. The females require the dense proteins and iron found in blood exclusively to synthesize and mature their eggs.
And after taking a blood meal, she finds a stagnant pool of water to lay those eggs.
And the aquatic life cycle, progressing from egg to wriggling larva to pupa and finally emerging as a flying adult, takes roughly 5 to 14 days, heavily dependent on ambient temperature and rainfall.
Now, there are over 400 species of Anopheles mosquitoes out there, but the epidemiological data indicates only about 30 or 40 of these are major drivers of human malaria. So what separates a lethal vector from just a nuisance?
This requires distinguishing between vector competence and vectorial capacity. They sound similar, but in public health, the distinction is profound.
Okay, lay it out for us.
Vector competence is a strict binary biological metric. It simply asks, does this specific mosquito species possess the genetic and physiological ability to become infected by Plasmodium, support its development, and secrete sporozoites?
So just the biological hardware. Can it harbor the pathogen?
Precisely. Many mosquitoes are completely refractory. Their innate immune systems destroy the eukaryote before it can ever form a cyst. But vectorial capacity is the epidemiological math.
It includes behavior.
Yes, capacity incorporates competence, but it multiplies it by behavioral and ecological variables. What is the density of the mosquito population? What's their biting rate? What's their host preference? And most critically, what is their daily survival probability?
So you could have a mosquito species that is 100% biologically competent, but if it overwhelmingly prefers to bite birds,
Or if its average lifespan is only 4 days, its vectorial capacity for human malaria is effectively zero.
we really need to drill down into those behavioral preferences because these traits dictate exactly which public health interventions will succeed or fail. We categorize vectors by who they bite, and where they rest.
Let's start with host preference. Mosquitoes rely on chemical trails, carbon dioxide, lactic acid to find a blood meal. Anthropophilic vectors have evolved a strong preference for humans.
While zoophilic vectors prefer animals, like cattle.
Right. In an area dominated by zoophilic mosquitoes, human transmission is naturally lower because the bites are wasted on dead-end animal hosts.
Then we categorize them by feeding location. Endophagic mosquitoes prefer to hunt and feed indoors, flying into houses at night. Exophagic mosquitoes prefer to feed outdoors.
And finally, the resting location, which is crucial for interventions. Endophilic mosquitoes, after taking a heavy blood meal, are sluggish. They look for the nearest vertical surface to rest and digest.
Which is usually the interior wall of the house they just fed in.
Exactly. Whereas exophilic mosquitoes take their blood meal and immediately fly back out the window to rest in the surrounding vegetation.
Okay, so when you combine these behavioral metrics, you start to see why sub-Saharan Africa bears such a disproportionate, devastating burden of global malaria mortality. It really comes down to one specific vector, doesn't it? Anopheles gambia.
Anopheles gambiae is an evolutionary nightmare for human health. It is the biological perfect storm.
How so?
It is highly anthropophilic. It vastly prefers human blood. It is intensely endophagic. It actively seeks out human dwellings at night. It is strongly endophilic. It rests on interior walls.
Check, check, check.
And it's an opportunistic breeder, laying eggs in tiny, temporary, muddy footprints. And crucially, it has a relatively high daily survival rate, meaning a large proportion live past that 10 to 18-day incubation period.
Wow. So it is a highly efficient, long-living assassin that breaks into your home, feeds exclusively on you, and then comfortably rests on your bedroom wall while the parasite incubates inside it.
That resting behavior, as we'll discuss later, is the exact vulnerability that indoor insecticide spraying targets.
Right. But before we leave transmission dynamics, I have to play devil's advocate regarding the vector. We're building this massive framework around mosquitoes. But in modern clinical medicine, does this disease always require the insect? Are there mechanical workarounds?
It is statistically rare, but biologically possible because the infectious agents, the merozoites, reside directly inside the red blood cells. Any mechanism that transfers infected red blood cells from one human to another bypasses the mosquito entirely.
So, like contaminated blood transfusions.
Transfusions, organ transplantation, and the sharing of needles among intravenous drug users.
And there is vertical transmission as well, right?
Yes, congenital malaria. If a mother has a heavy parasite burden in her placenta, the mechanical barrier can occasionally fail, particularly during delivery, allowing infected red blood cells to cross into the fetal circulation.
So the infant is born already suffering from an active blood stage infection.
Yes. And we must also mention the phenomenon of airport malaria.
Oh, this is fascinating.
It occurs when a highly infectious, long-lived vector like an Anopheles gambio from West Africa gets trapped in the cargo hold or cabin of an intercontinental flight.
And it just hitches a ride to Europe.
It survives the journey, escapes near an airport in a non-endemic city like Paris or London, and bites a local resident who has zero travel history. It is a terrifying diagnostic puzzle for the local physician.
I can imagine. But practically speaking, for 99.9% of the global burden, the vector is the bridge. But the vector is only dangerous if the human host is vulnerable, which leads us to the epidemiology of risk.
Host, environment, and their interaction.
Exactly. because two people can be bitten by the exact same infected mosquito, and one will develop a mild fever while the other progresses to fetal brain swelling.
And that divergence in clinical outcome is primarily driven by immunity. In areas of highly stable, intense malaria transmission, human beings are exposed to infectious bites dozens or even hundreds of times a year, starting from birth.
Right, relentless exposure.
This relentless exposure forces the human immune system to adapt. By late childhood, survivors develop what is known as partial or semi-immunity.
It's crucial to define partial here. It doesn't mean they never get infected, right?
Exactly. It means they tolerate the infection. Their immune system allows a low level of parasitemia to persist in the blood without triggering that massive systemic cytokine storm.
So an adult living in a highly endemic region might have active falciparum swimming in their blood right now, but they feel completely fine. They go to work, they farm, they have zero fever.
But that partial immunity is incredibly fragile. It requires constant reminders from incoming infectious bites to maintain the protective antibody titers.
So if they move away,
If that asymptomatic adult leaves the endemic region and moves to a country without malaria for just a few years, their immunity wanes rapidly. If they return to their home village for holiday, they are functionally naive again.
an infection that would have been completely asymptomatic 3 years ago can now rapidly progress to severe, life-threatening disease.
Yes.
This immunity framework really clearly defines who dies from this disease. If repeated exposure builds immunity, then the absolute highest risk group must be those who haven't lived long enough to be repeatedly exposed.
Exactly. Children between the ages of 6 months and 5 years bear the overwhelming brunt of global malaria mortality.
Why 6 months? What protects them before that?
For the first 6 months of life, infants are somewhat protected by maternal IgG antibodies that cross the placenta, as well as the high concentration of fetal hemoglobin in their blood, which the parasite struggles to consume.
But around 6 months, those maternal antibodies degrade.
The child is suddenly left completely defenseless against a pathogen they have absolutely no immunological memory of.
They are a blank slate facing a devastating pathogen. But immunity isn't just acquired, is it? The story of malaria is deeply intertwined with human genetics.
The evolutionary pressure exerted by this single microscopic parasite has been so intense over so many millennia that it has permanently rewritten the human genome in endemic areas.
We see this most famously with hemoglobinopathies, like the sickle cell trait.
Right. We all remember learning about the sickle cell trait in early genetics courses. The sickle cell gene mutation causes a single amino acid substitution in the beta-globin chain.
And if you inherit 2 copies homozygous HBSS, you develop sickle cell disease.
Which is a devastating condition characterized by painful vaso-occlusive crises and a severely shortened lifespan. From a pure evolutionary standpoint, a gene that causes such a lethal disease should have been aggressively bred out of the human population thousands of years ago.
But it hasn't been. In massive swaths of sub-Saharan Africa, the gene is highly prevalent. And the reason is the heterozygous advantage.
If you inherit one normal hemoglobin gene and one mutated sickle gene heterozygous HBAS, known as the sickle cell trait, you do not suffer from the severe vaso-occlusive crises of sickle cell disease.
Okay, so you're mostly healthy.
But your red blood cells contain just enough abnormal hemoglobin to make life incredibly difficult for Plasmodium falciparum. The parasite requires a highly stable, structured red blood cell to remodel the actin's cytoskeleton and build its own transport pathways.
So the sickle trait messes up the parasite's renovation plans.
Exactly. The altered structure and increased oxidative stress inside an HBAS red blood cell severely inhibit the parasite's ability to replicate. The presence of that one mutated gene confers massive, life-saving protection against severe, fatal falciparum malaria.
The evolutionary math is brutal but clear. The protection against malaria is so profound that it keeps the sickle cell gene circulating at high frequencies in the population, despite the tragic cost to those who inherit 2 copies.
And it isn't just the shape of the hemoglobin. The evolutionary arms race extends to the actual surface receptors on the red blood cell.
Right. We discussed earlier how Vivax is the most widely distributed malaria species globally, found across South America, South Asia. But if you look at an epidemiological map of West Africa, Vivax is almost non-existent. There's this massive geographical void.
That void is purely genetic. To physically enter a human red blood cell, Plasmodium vivax relies on a specific lock and key mechanism. It uses a protein called the Duffy binding protein, to attach to a receptor on the surface of our red blood cells called the Duffy antigen.
And if you lack that Duffy antigen,
The Vivax parasite simply bounces off the cell. It cannot invade and it cannot cause disease. Over thousands of years, the intense pressure of Vivax infection drove a genetic bottleneck.
so today, the vast majority of the indigenous population in West Africa is Duffy negative. The human population literally changed the cellular locks to lock the parasite out.
It's an incredible adaptation.
It really is. Now, beyond genetics and acquired immunity, we have to address pregnancy because pregnancy fundamentally alters a woman's physiological and immunological baseline. The Oxford textbook emphasizes that pregnant women are a uniquely vulnerable demographic.
Yes, even if they had previously acquired strong partial immunity. Pregnancy induces a state of relative immunosuppression to prevent the mother's body from rejecting the genetically distinct fetus.
Right, the immune system has to dial down.
Consequently, pregnant women lose a significant degree of their acquired anti-malarial immunity, particularly during their first and second pregnancies. They are far more likely to develop severe clinical symptoms, high parasite densities, and profound maternal anemia.
And there is a devastating, highly specific mechanism at play here involving the placenta, right? Placental sequestration. How does that work mechanically?
This is one of the most insidious adaptations of Plasmodium falciparum. The parasite modifies the surface of the infected red blood cell to express a protein VR2CSA that binds exclusively to chondroitin sulfate A.
Which is found where?
It's a carbohydrate found almost entirely in the placenta. The parasitized red blood cells act like magnets, flocking to and tethering themselves inside the intervillous space of the placenta.
Wow.
They physically hide out there, rapidly multiplying and choking off the maternal blood supply to the developing fetus.
This creates a terrifying diagnostic blind spot. Because the parasites are sequestered deep in the placental tissue, a clinician could draw peripheral blood from the mother's arm, look at it under a microscope, and see absolutely zero parasites.
The blood smear is negative, but the placenta is packed with them, and the clinical consequences are catastrophic. The mother suffers severe anemia, and the restriction of blood flow and nutrients leads to intrauterine growth restriction, preterm delivery, and low birth weight.
And in endemic regions, malaria in pregnancy is a massive hidden driver of infant mortality because a low birth weight baby is exceptionally vulnerable to all other infections in their first year of life. So we've mapped out the host vulnerabilities. But the environment dictates how aggressively the vector can exploit those vulnerabilities. The environment sets the baseline rules of the chess match.
Ambient temperature is the primary dial. If the weather cools, the extrinsic incubation cycle inside the mosquito lengthens. If it drops too low, the mosquito will die of old age before the parasite ever reaches her salivary glands.
Which is why altitude serves as a natural barrier. High mountain regions are generally malaria-free.
Conversely, as temperatures rise, the parasite's incubation cycle accelerates. A process that takes 14 days at a cool temperature might take only 10 days in extreme heat, drastically increasing the mosquito's infectious lifespan.
And rainfall is the other massive variable, providing the physical aquatic habitat for the larvae. Though the textbook points out a fascinating paradox regarding rain.
Oh, right.
While you need rain to create the breeding pools, a massive torrential monsoon downpour can actually temporarily halt transmission by violently flushing the larvae out of their breeding sites and killing them.
To capture all this environmental and entomological chaos in a single, quantifiable metric, epidemiologists use the entomological inoculation rate, or EIR.
The EIR, how is that calculated?
It's the mathematical synthesis of the hazard. It is calculated by taking the human biting rate, the average number of mosquito bites a person receives per night, and multiplying it by the sporozoa rate, which is the fraction of those mosquitoes that actually have infectious sporozoites in their salivary glands.
And the EIR gives you an astonishingly clear picture of the risk. An EIR of 1 means you get 1 infectious bite per year. But in some hyper-endemic parts of rural sub-Saharan Africa,
the EIR can exceed 500.
500. That means a young child is receiving more than 1 infectious parasite-loaded mosquito bite every single night of the year.
When you conceptualize an EIR of 500, the inevitability of infection becomes obvious, and when that exposure inevitably leads to infection, the battleground shifts instantly from the physical environment to the clinical environment.
Which brings us to the clinical manifestations and diagnosis. Let's walk through the timeline. A patient receives an infectious bite. They don't get sick the next day.
No, there is an incubation period that correlates with that liver phase we discussed earlier. Typically, this incubation period lasts between 12 and 35 days, with falciparum leaning toward the shorter end, averaging around 12 days.
And during this entire time, the patient feels perfectly fine.
But when those massive liver schizonts rupture and the merozoites hit the bloodstream, the symptoms begin. And the true danger of early malaria is that the presentation is notoriously, frustratingly nonspecific.
It mimics almost every other viral or bacterial syndrome. A low-grade fever, malaise, a dull headache, muscle aches, some nausea. In a Western clinic, it looks like a seasonal flu.
And in a tropical clinic, it looks like early dengue or typhoid. If a physician doesn't explicitly ask for a travel history or doesn't have a high index of suspicion in an endemic zone, it is incredibly easy to miss.
And missing early falciparum malaria is disastrous because the progression from uncomplicated to severe disease can happen in a matter of hours.
We differentiate the clinical state strictly into uncomplicated and complicated or severe malaria. Uncomplicated means they have those flu-like symptoms, they are febrile, but their vital organs are functioning normally and they can tolerate oral medications. Their parasitemia is usually low, maybe under 1%.
But if it's falciparum and it is left untreated, that parasitemia rapidly multiplies. Once it crosses certain thresholds or once organ systems begin to fail, we classify it as severe malaria.
This is an absolute high-stakes medical emergency. We are talking about severe anemia, acute respiratory distress, spontaneous bleeding, and shock.
I want to drill deeply into the why here. Why does falciparum cause such devastating multi-organ failure compared to Vivax or Oval? The textbook contributes this to a process called cytoadherence. Can we unpack the biophysics of what that actually means?
Cytoadherence is the defining pathophysiological weapon of Plasmodium falciparum. As the parasite matures inside the red blood cell, it fundamentally re-engineers the cell's architecture. It exports specific parasite proteins, most notably one called PFEMP1, out to the surface of the red blood cell membrane.
And what do these proteins do?
They form tiny, sticky knobs on the outside of the cell. These knobs act like molecular Velcro, binding tightly to specific receptors like CD36 or ICAM1, which are located on the endothelial cells lining our small blood vessels and capillaries.
So the infected red blood cells intentionally anchor themselves to the walls of the blood vessels. Why would they do that?
It's an evasion tactic. The human spleen is a massive filter designed to identify and destroy abnormal or infected red blood cells. By tethering themselves to the walls of the capillaries and the deep tissues, the parasitized red blood cells avoid circulating through the spleen. They hide in the microvasculature.
That's a brilliant survival strategy for the parasite, but it is catastrophic for the human host because you suddenly have millions of red blood cells stopping dead in the tiny capillaries of your brain, your kidneys, your lungs.
They act like microscopic traffic jams. Microvascular congestion. The blood flow slows to a halt. This completely starves the surrounding tissue of oxygen, leading to localized hypoxia, tiny ischemic infarcts, and capillary leakage.
And when this profound cytoadherence happens in the tiny blood vessels of the brain, the result is cerebral malaria.
Yes. The patient develops a rapidly progressive encephalopathy. They become delirious, suffer intractable seizures, and slip into a deep coma. If you look into their eyes, you will frequently see retinal hemorrhages caused by the occluded vessels.
Untreated cerebral malaria is almost uniformly fatal, isn't it?
Almost uniformly. And even with aggressive treatment, survivors, particularly young children, often suffer permanent neurological deficits, such as cortical blindness, epilepsy, or cerebral palsy.
And isn't just the brain. The textbook details a terrifying renal complication known historically as blackwater fever.
Blackwater fever is a manifestation of massive, sudden intravascular hemolysis. For reasons we still don't entirely understand, vast numbers of parasitized and unparasitized red blood cells suddenly burst simultaneously in the bloodstream.
Oh, wow. Just an explosion of red blood cells.
Exactly. This massive explosion dumps huge quantities of free hemoglobin into the plasma. The kidneys attempt to filter this toxic load, but the hemoglobin essentially clogs the renal tubules, leading to acute tubular necrosis and sudden renal failure.
And the high concentration of oxidized hemoglobin and malarial pigment in the urine turns it a dark, port wine or black color, which gave the syndrome its ominous name.
Alongside the organ failure, severe malaria frequently presents with profound metabolic derangements. Severe hypoglycemia is common, driven both by the parasite relentlessly consuming the host's glucose and by a hyperinsulinemic response.
And metabolic acidosis, a buildup of lactic acid due to the tissue hypoxia we discussed earlier, is one of the strongest clinical predictors of a fatal outcome.
Now, before we move to diagnosis, we must draw a hard clinical line between two terms that are often confused but mean completely different things regarding treatment failure: recrudescence versus relapse.
Let's define them clearly.
Recrudescence occurs within days to weeks of treatment. It means the initial therapy failed to completely clear the blood vault. Whether due to an inadequate drug dose, poor patient adherence, or emerging drug resistance, a small population of parasites survived the treatment in the bloodstream.
So the infection was suppressed but never completely cured.
Exactly. Once the drug washes out of the system, those survivors rapidly multiply and the clinical symptoms return, whereas relapse operates on a completely different timeline.
Relapse happens weeks, months, or even years later, exclusively in Vivax or oval infections, right?
Right. The initial treatment was entirely successful at clearing the bloodstream. The patient was cured of the blood stage infection. But the treatment did not kill the dormant hypnozoites sleeping in the liver. When those sleeper cells eventually awaken and flood the bloodstream with a new wave of merozoites, that is a true relapse.
So to avoid both recrudescence and relapse, you have to know exactly which species you are fighting. How do we confirm the diagnosis? How do we see the enemy?
The historical and continuing gold standard is light microscopy. A clinician takes a drop of the patient's blood, creates both a thick and a thin smear on a glass slide, stains it with a specialized GIMSA dye, and examines it under a high-powered microscope.
The thin smear leaves the red blood cells intact, allowing a highly trained technician to analyze the specific morphology to identify the exact species, right?
And the thick smear lyses the red blood cells, concentrating the parasites to allow the technician to quantify the density of the infection, the parasitemia percentage.
It's a phenomenal diagnostic tool, but it has massive logistical bottlenecks. Microscopy requires a reliable supply of electricity, well-maintained equipment, high-quality stains, and crucially, an experienced microscopist who can spend 15 to 20 minutes meticulously scanning a single slide.
Which is really tough in a rural setting. If you were running a rural clinic in sub-Saharan Africa with no grid power, seeing 200 febrile children a day during the rainy season, relying exclusively on microscopy is operationally impossible.
Which is exactly why the introduction of rapid diagnostic tests, or RDTs, completely revolutionized global malaria control.
RDTs bypass the need for electricity and microscopes. They are simple, lateral flow chromatographic tests, visually identical to an at-home pregnancy test or a rapid COVID-19 antigen test. You place a drop of blood on the cassette, add a buffer solution, and wait 15 minutes.
How exactly are they detecting the parasite without seeing it?
They utilize monoclonal antibodies embedded in the test strip that bind to specific parasite antigens. Most tests detect histidine-rich protein 2, or HRP2, which is an antigen exclusively produced by Plasmodium falciparum, and many also detect parasite lactate dehydrogenase, PLDH, an enzyme produced by all the other human Plasmodium species.
So a single test cassette can tell a rural health worker instantly if the patient has a lethal falciparum infection or a non-falciparum infection.
Yes. There have been historical debates about the sensitivity of these rapid tests compared to a trained microscopist, particularly with low-density infections. Early generations had stability issues in extreme tropical heat, but modern RDTs are highly sensitive.
However, the parasite is fighting back, isn't it?
We are now seeing a deeply concerning biological adaptation in regions like the Amazon basin and parts of East Africa. The falciparum parasite is mutating and actually deleting the gene that produces the HRP2 antigen.
Wait, it just stops making the protein?
It stops producing the protein the test is looking for. So a patient can have a massive life-threatening falciparum infection, but the rapid test will show up completely negative. This forces us to constantly evolve our diagnostic tools to stay ahead.
And getting that diagnosis absolutely correct is the strict prerequisite for our next framework.
Yeah.
Treatment and management, because the moment the lab confirms the species, you are locked into a highly specific pharmacological pathway.
And the rules for prescribing these drugs are heavily dictated by evolutionary biology and global resistance patterns.
Let's break it down by severity and species. Let's start with uncomplicated Plasmodium falciparum. You cannot use the older drugs like chloroquine anymore.
No, the parasite has evolved widespread global resistance to chloroquine. Today, the absolute non-negotiable standard of care directed by the WHO is the use of artemisinin combination therapies, or ACTs.
I have to play devil's advocate here with ACTs.
Hmm.
The fundamental concept is polypharmacy, combining two very different drugs into one pill. You take a powerful, fast-acting artemisinin derivative, and you pair it with a slower-acting partner drug like mefloquine or lumefantrine.
Right.
In resource-poor settings where counterfeit drugs are rampant and patient compliance is difficult, isn't prescribing a complex combination therapy riskier? Why not just hit the patient with a massive single dose of artemisinin? It clears the parasite faster than any other molecule we have ever discovered.
You'd think a single powerful drug would be better, but the evolutionary math tells a completely different story. It comes down to half-lives and the raw number of parasites in the body.
Okay, walk me through the numbers.
An adult with a moderate falciparum infection might have a total parasite burden of 1,010,000 to the power of 12 parasites inside them. Artemisinin is a molecular buzzsaw. It reduces the parasite biomass by a factor of 10,000 in a single 48-hour cycle. It drops that trillion down drastically, and the patient feels instantly better. But artemisinin has an incredibly short half-life. It metabolizes and vanishes from the bloodstream in a matter of hours.
So it hits hard, but it leaves the battlefield almost immediately.
Exactly. If you only use artemisinin monotherapy, it eliminates 99.9% of the parasites. But because it leaves the body so quickly, it leaves behind maybe 100,000 surviving parasites.
And those survivors are the toughest ones.
By definition, they're the hardiest, most resilient members of the population. Without the drug present to suppress them, they rapidly multiply. The infection recrudesces, but this time, the entire population descends from parasites that survived artemisinin. You have actively selected for and bred artemisinin-resistant malaria.
So it is a forced evolutionary pressure. Monotherapy is essentially a training program for the parasite.
Precisely. This is why we absolutely must combine it with a partner drug. The artemisinin violently knocks out the vast majority of the parasite load in the first 2 days, rapidly resolving the patient's dangerous clinical symptoms.
And then the partner drug comes in.
The partner drug, which has a much longer half-life and stays in the bloodstream for weeks, acts as a biological mop. It hunts down and eliminates those final 100,000 stragglers, ensuring that absolutely zero parasites survive the encounter. ACTs are a dual-strike strategy engineered specifically to protect the longevity of the drugs themselves.
That is brilliant systems thinking applied to pharmacology. Now, what happens if the diagnosis is non-falciparum? Let's say the RDT comes back positive for Plasmodium vivax. Do we still use ACTs?
In many areas, ACTs are increasingly used for Vivax as well. However, in regions where Vivax remains sensitive to older drugs, chloroquine is still a highly effective and cheaper option for clearing the blood stage infection. Chloroquine concentrates inside the parasite's food vacuole and poisons it.
But as we established in the life cycle framework, clearing the blood is only half the battle with Vivax. You still have the hypnozoates securely locked away in the liver vault.
The sleeper cells. Chloroquine doesn't touch the liver. To achieve a radical cure, meaning you eradicate the blood infection and you completely sterilize the liver to prevent future relapses, you must administer a separate drug called primaquine.
Primaquine specifically targets and kills those dormant hypnozoites. But this introduces a massive, life-threatening clinical safety caveat, doesn't it?
Yes. You cannot simply hand a patient a course of primaquine and send them home. You must first test their blood for a specific genetic enzyme deficiency. Glucose 6-phosphate dehydrogenase, or G6PD deficiency.
We need to unpack the biochemistry of this because this is a classic board exam trap and a very real clinical hazard. What is the physiological mechanism connecting an antimalarial drug to a genetic enzyme deficiency?
G6PD is an enzyme that plays a critical role in protecting red blood cells from oxidative stress. It helps regenerate a molecule called NADPH, which in turn keeps glutathione in its reduced state. Reduced glutathione acts like a cellular fire extinguisher, neutralizing damaging reactive oxygen species.
And primaquine.
Primaquine is a drug that induces intense, massive oxidative stress inside the red blood cell.
It's essentially throwing biochemical matches into the cell.
Yes. In a normal patient with adequate G6PD, the enzyme rapidly regenerates the glutathione, puts out the oxidative fires, and the red blood cell survives just fine while the primaquine kills the parasite in the liver.
But if a patient is genetically deficient in G6PD?
Their red blood cells cannot regenerate glutathione. When you give them primaquine, the oxidative stress goes unchecked. The hemoglobin inside the cell denatures, forming rigid clumps called Heinz bodies.
And the spleen sees that and attacks it.
The spleen recognizes these damaged cells and aggressively rips them out of circulation. The result is massive, potentially fatal intravascular hemolysis. The patient's red blood cells literally pop and are destroyed.
So in attempting to cure a dormant liver infection, you could cause catastrophic iatrogenic anemia and acute renal failure.
Which is why G6PD testing prior to primaquine administration is an absolute clinical mandate.
Wow. Okay, now what happens when the frameworks fail? When the patient presents late or the initial treatment fails and they develop severe, complicated malaria. We are out of the rural clinic and into the intensive care unit. The patient is comatose or heavily acidotic. How does the protocol change?
Severe malaria dictates immediate hospitalization and a shift from oral to intravenous or intramuscular medication. You need maximum drug concentration in the blood immediately.
Historically, that was intravenous quinine, right?
Yes, 4V quinine was the gold standard. But massive multi-center global clinical trials, specifically the Aquamat and Sequomat trials, conclusively proved that intravenous artesunate is vastly superior.
How much better is it?
4V artesunate clears the parasite mass significantly faster and, most importantly, reduces overall mortality by almost a quarter compared to quinine. Today, 4V artesunate is the definitive preferred frontline treatment for severe malaria globally. Once the patient stabilizes and can swallow, you transition them back to a full oral course of ACTs to ensure complete clearance.
We have the rapid diagnostic tools. We have the sophisticated combination therapies. We understand the vector ecology. But to truly grasp why global health organizations allocate billions of dollars annually to this fight, we have to comprehend the sheer scale of the devastation.
We need to look at Section 7 of the Oxford Framework, the global burden of malaria.
And this is where the textbook forces us to confront some deeply conflicting, uncomfortable data.
The epidemiological data on malaria morbidity and mortality is staggering, but is also highly contested. It perfectly illustrates the immense difficulty of conducting global health surveillance in resource-poor environments.
Let's look at morbidity first, the raw number of clinical cases. The World Health Organization estimated roughly 219 million total cases of malaria worldwide in 2010.
However, a major independent research consortium known as the Malaria Atlas Project, or MAP, utilized advanced spatial modeling and estimated roughly 451 million clinical cases of Plasmodium falciparum alone in 2007.
That is a discrepancy of over 200 million cases. And if we visualize the MAP data, the textbook provides this chilling map demonstrating falciparum endemicity. You see an almost unbroken wall of dark gray shading across the vast majority of sub-Saharan Africa,
indicating intense, stable transmission where more than 40% of the population is infected.
But the map isn't just Africa. You see vast swaths of significant risk across the Indian subcontinent, Southeast Asia, and the Amazon basin in South America.
And looking at that geographical spread highlights exactly where the data diverges. Take India, for example. The WHO estimates for clinical malaria attacks in India were less than a third of the lower limit of the estimates produced by the MAP group.
Or consider Indonesia. The WHO estimated approximately 2 million clinical cases, but the MAP spatial models estimated 12 million falciparum cases. And that doesn't even account for the massive Vivax burden there.
The mortality data is equally divergent. The WHO estimated roughly 660,000 deaths in 2010, heavily concentrated among children under 5 in Africa. But the Institute for Health Metrics and Evaluation, the IHME, published their own estimate of 1.2 million deaths, nearly double the WHO figure.
What is particularly fascinating from an epidemiological standpoint is where the IHME models found those extra 500,000 deaths. While both organizations agreed that the vast majority of mortality occurred in Africa, the IHME models suggested a massively higher burden of mortality in older individuals.
Yes, they estimated over 400,000 deaths in people 5 years of age and older compared to just 91,000 estimated by the WHO for that same demographic.
As clinical professionals trying to understand the actual burden, what do we make of these wildly divergent numbers? How can the world's leading epidemiological organizations be hundreds of thousands of deaths and hundreds of millions of cases apart? Who is right?
Well, it's not necessarily about who is right. It's about the inherent limitations of the data sources. The regions bearing the highest burden of malaria are, by definition, often the most resource-poor settings with fragile or non-existent health infrastructure.
So routine laboratory confirmation of every fever simply does not happen.
Hundreds of thousands of deaths occur at home, in rural villages, completely unrecorded by any formal medical surveillance system.
So how do they estimate deaths if there are no hospital records?
They frequently rely on a tool called a verbal autopsy. Epidemiologists interview the grieving families months after a death, asking them to recall the specific symptoms their loved ones suffered before dying. Did they have a high fever? Did they have convulsions?
But as we discussed, severe malaria perfectly mimics bacterial sepsis, meningitis, and severe pneumonia. A verbal autopsy cannot differentiate a cerebral malaria death from a meningococcal meningitis death.
Exactly. So the models have to extrapolate from incredibly sparse, noisy data. The WHO models tend to be more conservative, heavily weighting clinical data. The MAP and IHME models rely more heavily on spatial transmission modeling and mathematical extrapolations.
But while the specific estimates differ dramatically, the fundamental conclusion is identical across every source. The burden of disease is almost unfathomably immense.
And focusing solely on the immediate mortality completely misses the massive hidden burden of the disease. We discussed the insidious impact of placental sequestration during pregnancy. The textbook estimates between 75,000 and 200,000 infant deaths annually are purely attributable to malaria-induced low birth weight.
Those infants don't technically die of malaria, they die of diarrhea or pneumonia because the malaria starved them in the womb.
We must also account for chronic disability. The Global Burden of Disease study calculated over 4 million years lived with disability. This is primarily driven by the debilitating, profound chronic anemia that repeated malaria infections cause.
A child who is chronically anemic cannot concentrate in school. An adult who is chronically anemic cannot physically labor in the fields. And this leads directly to the macroeconomic cost.
The direct costs for anti-malarial treatment and hospitalization are estimated at over $12 billion annually.
But the textbook points out a much darker economic reality. When you look at the macroeconomic data, the true cost isn't just the $12 billion in treatment. The true cost is measured in lost potential.
Countries with intensive, uncontrolled malaria transmission have per capita income levels that are only 33% of those countries without malaria.
The disease acts as an insurmountable anchor on a nation's economic growth. It traps entire generations in poverty by slashing agricultural productivity, draining household savings for medical care, and stunting educational attainment.
Which brings us to the ultimate public health challenge. If we understand the agent's biology, the vector's behavior, the host's genetic vulnerabilities, and the staggering macroeconomic reality, how do we engineer a systemic response to stop it?
We need to build a framework for prevention, control, and system impact. I think the most useful way to conceptualize this is through stacking interventions. No single tool is perfect.
We have to combine upstream engineering, trying to eliminate the threat before it ever reaches the human, with downstream armor, which protects the individual when the upstream systems fail.
Let's start with the cornerstone of upstream prevention, vector control. Specifically, insecticide-treated nets, or ITNs.
ITNs, and more specifically long-lasting insecticidal nets, or LLINs, have been the single most effective tool deployed in the last 20 years.
How do they work over the long term?
The pyrethroid insecticide is chemically bound directly into the polyethylene fibers of the net, meaning it survives years of use and multiple washings. They are a phenomenal multi-layered defense. They obviously act as a physical barrier, preventing the anophagic mosquito from reaching the sleeping child.
But more importantly, the insecticide actively repels or kills the mosquito upon contact.
So it isn't just protecting the child under the net, it is actively reducing the overall vector density and, crucially, reducing the vector's lifespan in the community.
The textbook notes that widespread deployment of ITNs has been proven to reduce overall childhood mortality, not just malaria mortality, but all-cause childhood mortality by staggering 17%.
The second major vector control pillar is indoor residual spraying, or IRS. This involves deploying teams to systematically spray a long-lasting insecticide formulation onto the interior walls and ceilings of human dwellings.
But this is where the entomological framework we built earlier is absolutely critical. IRS is an incredibly powerful tool, but it is only effective if the local malaria vector is predominantly endophilic.
Right, the mosquito has to actually land on the wall for the poison to work.
Exactly. If the vector is Anopheles gambiae, which loves to rest on indoor walls after a blood meal, IRS will decimate the mosquito population.
The mosquito feeds, lands on the wall to digest, absorbs the insecticide through its tarsi, and dies before the 14-day extrinsic incubation period finishes.
But if you spend millions of dollars deploying an IRS campaign in a region where the primary vector is exophilic, meaning it takes a blood meal and immediately flies out the window to rest in the jungle canopy, the intervention will completely fail.
The mosquitoes will never touch the sprayed walls, public health interventions must be meticulously tailored to local vector behavior.
Okay, so nets and spraying target the adult mosquitoes that have already entered the human environment. What about going further upstream? Source reduction. Can we just eliminate the breeding sites?
Source reduction involves altering the physical environment to remove standing water-draining swamps, filling in ditches, managing irrigation systems. Historically, this was incredibly successful.
It was the primary strategy used to eliminate malaria in the United States, right?
Right.
And during the construction of the Panama Canal.
Yes. But it has severe limitations in much of the world today. As we noted, Anopheles gambiae can breed in a rain-filled hoofprint or a discarded plastic cup. You cannot realistically drain every puddle in the Amazon basin or across sub-Saharan Africa.
So if we can't drain the water, we have to treat the water. The textbook discusses biological controls, which is a fascinating frontier, using nature to fight nature.
We use larvicides like BTI, a natural toxin produced by a soil bacterium that specifically destroys the mosquito larva's gut but is harmless to humans and other wildlife. But the truly cutting-edge biological control involves a parasitic bacterium called Wolbachia.
I really want to understand the mechanism here. How does infecting a mosquito with a bacteria stop human malaria?
Wolbachia is a naturally occurring bacterium that infects many insect species, but usually not major disease vectors. Researchers have discovered that if you artificially infect Aedes or Anopheles mosquitoes with Wolbachia in the lab, it fundamentally alters the mosquito's biology.
It competes with the parasite, right?
Yes. The Wolbachia aggressively competes with the Plasmodium parasite for vital resources like cholesterol inside the mosquito cells. It also seems to upregulate the mosquito's basal immune system, increasing reactive oxygen species in the midgut.
The result is that the mosquito becomes highly refractory to the parasite.
The Plasmodium cannot develop into sporozoites. If you release these Wolbachia-infected mosquitoes into the wild, they interbreed and pass the Wolbachia to their offspring, slowly rendering the entire wild mosquito population incapable of transmitting the disease.
That is incredible upstream engineering. But we cannot rely purely on the vector. We must also deploy downstream armor to protect the most vulnerable human hosts directly with prophylactic drugs. The public health strategy here is intermittent preventive therapy, or IPT.
IPT utilizes the concept of proactive chemoprophylaxis. We don't wait for the patient to get sick. We administer curative doses of antimalarials at predefined intervals to clear asymptomatic infections before they become dangerous.
And the most critical application is IPTP, intermittent preventive therapy in pregnancy.
Pregnant women are given doses of sulfadoxine pyrimethamine, or SP, during their routine antenatal clinic visits, regardless of whether they feel sick or have a positive blood smear.
And the goal there is to clear out that hidden placental sequestration we discussed earlier, explicitly to prevent low birth weight and maternal anemia.
Correct. We also use IPTC, which is seasonal malaria chemoprevention for children. In regions like the Sahel and Africa, malaria transmission is highly seasonal, spiking violently during the 3 to 4 month rainy season.
So we proactively administer monthly courses of antimalarials to all children under 5 during those specific months, providing a chemical shield during the period of highest risk.
And of course, there is traditional travel chemoprophylaxis daily pills like atovaquone-preguanil or doxycycline for non-immune individuals traveling into endemic zones.
Which naturally leads to the ultimate downstream armor, the holy grail of infectious disease control, a vaccine. We eliminated smallpox with a vaccine. We nearly eradicated polio with a vaccine. Why has a malaria vaccine been such a frustrating, decades-long scientific struggle?
It comes down to the immense biological complexity of the agent. A virus like measles or polio is relatively simple. It has a handful of proteins for the immune system to target. Plasmodium is a massive, complex eukaryote with thousands of genes.
And multiple life stages, right?
Exactly. It has sporozoite, liver stage, blood stage, gametocyte, and it expresses entirely different surface proteins at each stage. Furthermore, it actively evades the immune system by constantly shifting those surface proteins and by hiding inside the liver and red blood cells where circulating antibodies cannot reach it.
The textbook highlights the most advanced candidate at the time, the RTS vaccine. How exactly does it attempt to overcome that complexity? What is it targeting?
RTSS is a recombinant protein vaccine that targets the very first moment of infection. It is engineered to produce high levels of antibodies against the circumsporozoite protein, or CSP, which coats the surface of the sporozoite injected by the mosquito.
The goal is to intercept and destroy the sporozoites in the bloodstream before they can ever reach the safety of the liver.
But the clinical trial data discussed in the text is sobering. It showed a pooled phase 2 efficacy of roughly 36% in reducing clinical episodes. And alarmingly, that protection waned over time, dropping significantly after just a few years.
Why does it wane so fast?
The waning efficacy highlights the sheer mathematical overwhelming force of the parasite in high transmission areas. The vaccine generates a high antibody titer, which effectively blocks the sporozoites initially, but over time, those antibody levels naturally drop.
And in a region where a child might receive hundreds of infectious bites a year, eventually, the sheer volume of incoming sporozoites overwhelms the fading antibody blockade.
A few sporozoites slip through to the liver, and the blood-stage infection begins. A vaccine with 36% efficacy is still an incredibly valuable tool that can save tens of thousands of lives, but it is not the magic bullet that smallpox was.
So public health requires stacking all these imperfect tools. You use ITNs to block the bite, IRS to kill the resting mosquitoes, source reduction to limit breeding, IPT to protect the vulnerable, and vaccines to prime the immune system. We have to squeeze the transmission cycle from every conceivable angle.
But even with a perfectly stacked defense, we're fighting a continuously adapting enemy. Which leads us to our final analytical framework, Section 9, Challenges to Elimination.
Because the history of malaria control is haunted by the fact that we actually tried to eradicate it globally once before, and we failed spectacularly.
Let's talk about the Global Malaria Eradication Program, launched by the WHO in 1955. It was a massive, optimistic global campaign, heavily reliant on mass indoor spraying with the insecticide DDT and the mass administration of chloroquine.
And for a brief window, it looked like it was working right. They eliminated malaria entirely in dozens of countries, including parts of Europe and the Americas.
But in much of the developing world, particularly Africa and South Asia, the campaign stalled and then collapsed into a tragic recrudescence. Cases roared back to pre-intervention levels and sometimes higher.
Why did it fail?
It failed due to a lethal combination of operational arrogance and biological inevitability. Operationally, the program was rigidly top-down and highly militarized. The textbook highlights a profound lesson here. The WHO forced highly trained research scientists and malariologists to act as logistics managers, overseeing fleets of spray trucks.
They completely bypassed local community health infrastructure. They didn't build rural clinics or train local doctors, they just sprayed chemicals.
When the initial funding inevitably dried up and the international spray teams packed up and went home, there was absolutely no local health system in place to maintain the surveillance or treat the straggling cases.
And because the transmission had been suppressed for a few years, the local population had actually lost whatever partial acquired immunity they previously had. So when the parasite returned, it hit a completely naive, unprotected population. It was an epidemiological disaster.
And while the operational logistics were failing, biological resistance was evolving in real time. We forced the chess pieces to move. We sprayed millions of tons of DDT, which exerted a massive evolutionary selection pressure on the mosquitoes.
The susceptible mosquitoes died, and the rare genetic mutants that possessed enzymes capable of metabolizing the insecticide survived and repopulated.
Today, the legacy of that arms race is terrifying. The textbook notes that 64 countries report widespread mosquito resistance to at least one major class of insecticide, particularly the pyrethroids that we rely on for our life-saving bed nets.
And it isn't just the mosquito mutating. The parasite is evolving to survive our best drugs.
We are currently tracking a true nightmare scenario, the emergence of Plasmodium falciparum strains that are actively resistant to artemisinin. This resistance first emerged, as it historically almost always does, along the Thai-Cambodian border.
I've always wondered about that geographical specificity. Why is the Thai-Cambodian border consistently ground zero for global anti-malarial drug resistance? It happened there with chloroquine, then with mefloquine, and now artemisinin.
It's a complex convergence of factors. You have intense seasonal transmission. You have high levels of human migration miners, loggers, refugees moving frequently across borders, making it impossible to ensure they complete a full course of medication.
But a massive systemic driver of this resistance is the unregulated illicit drug market, isn't it?
The textbook cites a horrifying, almost unbelievable statistic. In some studies, up to 36% of antimalarials collected in Southeast Asia were completely falsified or substandard.
36% counterfeit.
Yes. These fake drugs often contain just a tiny fraction of the active artemisinin ingredient. It is enough to temporarily clear the patient's fever and make them think they're cured, but it is nowhere near enough to kill the entire parasite biomass.
It subjects the parasite population to a subtherapeutic dose, which is the exact perfect recipe for selecting and breeding drug-resistant mutants.
Those counterfeit factories are essentially mass-producing drug-resistant malaria and seeding it into the global population.
So when we synthesize this entire landscape-deficient surveillance data, global funding plateauing around $1.8 billion annually when we need billions more, mosquitoes increasingly surviving our best insecticides, and the parasite actively evolving to survive our most powerful combination therapies. As public health professionals, is the concept of complete global eradication just a dangerous pipe dream?
It demands that we balance pragmatic realism with cautious optimism. The ecological, evolutionary, and political hurdles are absolutely monumental. We learned in the 1960s that we cannot just spray our way out of this with a top-down mandate.
But we are not empty-handed.
Exactly. The innovations we discussed, genomic surveillance to track resistance mutations in real time, Wolbachia-infected mosquito releases, highly advanced spatial epidemiological modeling, and the slow but steady progress on next-generation vaccines, these are incredibly powerful new pieces on the chessboard.
The science has reached a point where regional elimination is highly feasible and happening right now. But complete, permanent global eradication will require an unprecedented, sustained, multi-generational commitment to building robust local health systems in the poorest regions on Earth.
It is the ultimate test of our global health infrastructure.
Which brings us to a final provocative thought to leave you with. If human beings and Plasmodium have been locked in this ruthless genetic arms race for millennia, a war so intense it literally changed the shape of our red blood cells to survive, what happens next?
With climate change, you mean?
Exactly. We are currently living through unprecedented anthropogenic climate change. As global temperatures rise and rainfall patterns shift, the geographic habitat of the highly competent Anopheles mosquito is expanding into higher altitudes and higher latitudes.
It is moving into previously naive populations that have absolutely no genetic or acquired immunity.
How will that environmental shift rewrite the next chapter of human evolution and global health security? The chessboard is actively expanding.
And anticipating the parasite's next move on that expanding board is the defining challenge for the next generation of global public health leaders.
Before we go, let's lock in the core frameworks we built today with a few rapid recall prompts. We'll ask the question, pause for a moment to let you think, and then give the answer.
Sounds good.
Prompt 1. What is the specific pathophysiological mechanism driven by the PFEMP1 protein that leads to the severe complications and multi-organ dysfunction seen in Plasmodium falciparum infections?
I'll give you a second.
The answer is cytoadherence. Parasitized red blood cells develop sticky knobs that adhere to the endothelial lining of small blood vessels, avoiding splenic clearance but causing profound microvascular congestion and tissue hypoxia. Review the clinical manifestations section for more detail.
Okay, prompt 2. Which genetic trait provides almost complete resistance to infection by Plasmodium vivax and explains why this specific species is virtually absent in West Africa?
Take a moment.
The answer is being negative for the Duffy blood group antigen. The vivax parasite requires this specific receptor to physically invade the red blood cell. Evolutionary pressure has rendered most West African populations Duffy negative. This is detailed in the epidemiology and host susceptibility section.
Prompt 3. If a local entomological survey determines that the primary malaria vector in a region is strictly exophilic, which major heavily funded public health intervention will be largely biologically ineffective?
Think about the resting behavior.
The answer is indoor residual spraying, or IRS. Because exophilic mosquitoes prefer to rest outdoors in vegetation immediately after feeding, they will never come into contact with the insecticide sprayed on the interior walls of human dwellings. You can review this in the prevention and vector control section.
Prompt 4. A patient returns from Southeast Asia and is diagnosed with Plasmodium ovale. Before administering a full course of primaquine to clear the dormant liver hypnozoites and achieve a radical cure, what underlying genetic condition must you absolutely test the patient for?
This is a huge safety point.
You must test for glucose 6-phosphate dehydrogenase, or G6PD, deficiency. Administering primaquine to a G6PD deficient patient induces unchecked oxidative stress, triggering massive and potentially fatal intravascular hemolysis. This critical safety protocol is covered in the treatment and management section.
Prompt 5. What is the underlying evolutionary and epidemiological rationale for mandating the use of artemisinin-based combination therapies, or ACTs, rather than utilizing artemisinin as a monotherapy for uncomplicated falciparum malaria?
Why do we combine them?
The rationale is to actively actively prevent the emergence of anti-malarial drug resistance. The rapid-acting artemisinin violently reduces the bulk of the parasite mass but washes out quickly. The long-acting partner drug acts as a mop to eliminate the surviving fraction, ensuring no resilient mutants survive the treatment cycle. This is discussed heavily in the treatment and drug resistance section.
And with that, we wrap up our analysis of the board.
Here are the details of this episode.
Malaria. Byline. Vector-borne transmission, environmental determinants, and global elimination efforts. Or episode description. Malaria remains one of the most significant parasitic diseases worldwide, with a disproportionate burden in sub-Saharan Africa. Transmitted by the Anopheles mosquito, malaria reflects the complex interplay between environment, vector ecology, socioeconomic conditions, and health system capacity. This episode explores global epidemiology and regional distribution. This episode explores the life cycle of Plasmodium species. This episode explores transmission dynamics and seasonality. This episode explores high-risk populations, children under 5, and pregnant women. This episode explores insecticide-treated bed nets and indoor residual spraying. This episode explores artemisinin-based combination therapy. This episode explores drug and insecticide resistance. This episode explores climate change and vector expansion. This episode explores global eradication initiatives and surveillance. This episode explores malaria illustrates how disease control depends not only on treatment but on vector management, housing conditions, and sustained prevention strategies. Gains achieved over recent decades through bednet distribution, rapid diagnostic testing, and effective therapies have reduced mortality substantially, yet elimination remains uneven. This episode explores environmental change, urbanization, and resistance threaten progress, underscoring the need for continued investment and innovation. This episode explores malaria control is both ecological and political, shaped by geography, governance, and sustained commitment. This episode explores malaria remains a major cause of morbidity and mortality in endemic regions. This episode explores transmission depends on vector ecology and environmental conditions. This episode explores bed nets and indoor spraying significantly reduce risk. This episode explores drug and insecticide resistance threaten progress. This episode explores climate change may expand transmission zones. This episode explores that children and pregnant women are particularly vulnerable. This episode explores sustained global coordination is required for elimination.